Welcome back to Concepts of Biology. We'll read Chapter 3, Cell Structure and Function. Figure 3.1. Nasal sinus cells viewed with a light microscope, onion cells viewed with a light microscope, and Vibrio tasmaniensis bacterial cells viewed using a scanning electron microscope are from very different organisms, yet all share certain characteristics of basic cell structure. Chapter Outline. Section 3.1. How cells are studied. 3.2. Comparing prokaryotic and eukaryotic cells. 3.3. Eukaryotic cells. 3.4. The cell membrane. 3.5. Passive transport. 3.6. Active transport. Introduction. Close your eyes and picture a brick wall. What is the basic building block of that wall? It is a single brick, of course. Like a brick wall, your body is composed of basic building blocks, and the building blocks of your body are cells. Your body has many kinds of cells, each specialized for a specific purpose. Just as a home is made from a variety of building materials, the human body is constructed from many cell types. For example, epithelial cells protect the surface of the body and cover the organs and body cavities within. Bone cells help to support and protect the body. Cells of the immune system fight invading bacteria. Additionally, red blood cells carry oxygen throughout the body. Each of these cell types plays a vital role during the growth, development, and day-to-day maintenance of the body. In spite of their enormous variety, however, all cells share certain fundamental characteristics. 3.1. How cells are studied. By the end of this section, you will be able to describe the roles of cells in organisms, compare and contrast light microscopy and electron microscopy, and summarize the cell theory. A cell is the smallest unit of a living thing. A living thing, like you, is called an organism. Thus, cells are the basic building blocks of all organisms. In multicellular organisms, several cells of one particular kind interconnect with each other and perform shared functions to form tissues, for example, muscle tissue, connective tissue, and nervous tissue. Several tissues combine to form an organ, for example, stomach, heart, or brain, and several organs make up an organ system, such as the digestive system, circulatory system, or nervous system. Several systems functioning together form an organism, such as an elephant. There are many types of cells, and all are grouped into one or two broad categories: prokaryotic and eukaryotic. Animal cells, plant cells, fungal cells, and protist cells are classified as eukaryotic, whereas bacteria and archaea cells are classified as prokaryotic. Before discussing the criteria for determining whether a cell is prokaryotic or eukaryotic, let us first examine how biologists study cells. Microscopy. Cells vary in size. With few exceptions, individual cells are too small to be seen with the naked eye, so scientists use microscopes to study them. A microscope is an instrument that magnifies an object. Most images of cells are taken with a microscope and are called micrographs. Light microscopes. To give you a sense of the size of a cell, a typical human red blood cell is about 8 millionth of a meter or 8 micrometers in diameter. The head of a pin is about 2/1000ths of a meter in diameter. That means that approximately 250 red blood cells can fit on the head of a pin. The optics of the lenses of a light microscope changes the orientation of the image. A specimen that is right side up and facing right on the microscope slide will appear upside down and facing left when viewed through a microscope, and vice versa. Similarly, if the slide is moved left while looking through the microscope, it will appear to move right, and if moved down, it will seem to move up. This occurs because microscopes use two sets of lenses to magnify the image. Due to the manner in which light travels through the lenses, this system of lenses produces an inverted image. Binoculars and a dissecting microscope work in a similar manner, but include an additional magnification system that makes the final image appear to be upright. Most student microscopes are classified as light microscopes. Figure 3.2a. Visible light both passes through and is bent by the lens system to enable the user to see the specimen. Light microscopes are advantageous for viewing living organisms, but since individual cells are generally transparent, their components are not distinguishable unless they are colored with special stains. Staining, however, usually kills the cells. Light microscopes commonly used in the undergraduate college laboratory magnify up to approximately 400 times. Two parameters that are important in microscopy are magnification and resolving power. Magnification is the degree of enlargement of an object. Resolving power is the ability of a microscope to allow the eye to distinguish two adjacent structures as separate. The higher the resolution, the closer those two objects can be, and the better the clarity and detail of the image. When oil immersion lenses are used, magnification is usually increased to 1,000 times for the study of smaller cells, like most prokaryotic cells. Because light entering a specimen from below is focused onto the eye of an observer, the specimen can be viewed using light microscopy. For this reason, for light to pass through a specimen, the sample must be thin or translucent. A second type of microscope used in laboratories is the dissecting microscope, figure 3.2b. These microscopes have a lower magnification, 20 to 80 times the object size, than light microscopes, and can provide a three-dimensional view of the specimen. Thick objects can be examined with many components in focus at the same time. These microscopes are designed to give a magnified and clear view of tissue structure as well as the anatomy of the whole organism. Like light microscopes, most modern dissecting microscopes are also binocular, meaning that they have two separate lens systems, one for each eye. The lens systems are separated by a certain distance and therefore provide a sense of depth in the view of their subject to make manipulations by hand easier. Dissecting microscopes also have optics that correct the image so that it appears as if being seen by the naked eye and not as an inverted image. The light illuminating a sample under a dissecting microscope typically comes from above the sample, but may also be directed from below. Figure 3.2. Most light microscopes used in a college biology lab can magnify cells up to approximately 400 times. Dissecting microscopes have a lower magnification than light microscopes and are used to examine larger objects such as tissues. This ends Chapter 3, Section 3.1, Microscopy, Light Microscopes. In our next video, we'll read Electron Microscopes.